Okay, hi everyone. Um, let's continue talking about autoimmune diseases and once again try to understand how, number one, that autoimmunity is all about the immune system making mistakes and targeting self signals that should normally be tolerated. Right? And number two, how these types of mistakes um, follow the typical types of hypersensitivity models that we've been studying for allergies, as well as graft and tissue transplantation rejection. An important part of understanding autoimmunity, which we introduced last lecture, is understanding how genetics can fit into this. So let's go ahead and continue talking about that. So first off, let's check in and see where we are in the semester. I think it's all of you know we're in week 15 now, and so we're really nearing the end of our immunology course. So here we are, Wednesday, lecture 36. So we'll finish talking about autoimmunity today. I have these eyes here to remind you that you do have a quiz this week. Okay, the quiz is actually a combination of 5 questions from chapter 15, which was the grafts and transplantation stuff, and then 5 questions from the autoimmune stuff that we've been learning Monday and today. So I will make that quiz available right after lecture is done today, and I'll give you until Friday night to complete it. All right, that way you have a little extra time since we're just finishing with this new material today. So don't forget though. Okay, so I'll make that quiz available and Lalima should alert you that it's available. Keep in mind you have until Friday night. I'm always cautious about when I give you extensions because if you guys are like me, I get used to a real routine, like turning in quizzes Thursday night every week. And then suddenly if I get an extension, sometimes that messes me up, right? So don't get messed up. It's due Friday night because you have an extra day to do it. Then on Friday of this week, we'll talk about cancer. And Friday's lecture and Monday's lecture will really wrap up the course by looking at the role of the immune system in cancer. And you will have one last quiz on the chapter 17 cancer stuff that will be due normal time next week, Thursday night. All right. And then we'll review for the third exam on Wednesday. And then the third exam will be on Monday, the 10th at 12 noon, which is the time that's given to us for final exams, even though the exam itself is just a third midterm exam, just like the previous two. Okay, so be aware of those things. Make yourself a note, set an alarm or whatever, so you don't forget about these last couple quizzes that are coming up. Okay. We looked at this table last time as a nice way to introduce ourselves to different examples of autoimmune diseases and understand how they fit the type 2, type 3, and type 4 hypersensitivity models. So last time we looked at examples, for example, type 2, we looked at hemolytic anemia. We looked at lupus as an example of a type 3, and we looked at MS or multiple sclerosis as an example of a type 4, just to sort of get our feet wet and understand how certain things can happen to fit these different hypersensitivity models. So today, let's finish up by looking at the other examples that are on this table. So let's start off with the type 2, so Graves' disease and myasthenia gravis. Because it's a type 2 hypersensitivity, that means these diseases, Graves' disease and myasthenia gravis, you know that they involve antibodies, right? And type 2 hypersensitivity mostly involves IgGs. IgGs that your adaptive immune response produces, in this case, by mistake, right? That are somehow now targeting something in your body that they really shouldn't be. When we talk about antibodies, oftentimes we forget that antibodies are themselves proteins, right? They're expressed from the Ig locus, that set of genes, the VDJ recombination that puts together different fragments of genes to make one complete usable gene that then gets expressed as your light chain and your heavy chain, and then you have two light chains and two heavy chain peptides that come together to form your immunoglobulin, right? So the reason I say we oftentimes forget about that is because if Igs are themselves proteins, and antibodies, of course, are the free-floating forms of these Igs, then they can cause trouble just by the fact that they are proteins floating around that can interact with other proteins. Right? So in Graves' disease, these antibodies are causing trouble by interacting improperly with different receptor proteins that are on the surfaces of certain cells in your body. Remember, our cells are covered with different proteins that function as receptors, right? So these are proteins that are part of the membrane that are the right shape to be able to bind some other, usually small protein that comes floating around. And that little small protein acts as some sort of a signal. So the small little proteins that we've been studying all semester long are the various types of cytokine messages that our immune system is highly dependent on, right? Cytokine messages that work to communicate between cells the status of different things like infection or inflammation, etc. Right? So in your thyroid tissue, you have cells that have on their surface special proteins that are the thyroid stimulating hormone receptor. Okay, so in this cartoon here, these gray cells represent cells that are part of your thyroid tissues. And up in your brain, or at the base of your brain, you have your pituitary gland. And I think you guys know the pituitary gland makes all kinds of hormones, all kinds of signaling molecules that are used throughout the body to control things like metabolism, growth, etc. So in this cartoon, the pituitary gland is making this thyroid stimulating hormone. So it's a small molecule that then goes through your blood, and eventually some of that gets received by the TSH receptor protein on the surfaces of your thyroid tissues. And what that does is that stimulates these thyroid cells to go ahead and release thyroid hormones, right? So this is sort of an example of a hormone that's coming from your pituitary that's used as kind of like one of the master control hormones that then can cause other endocrine tissues or hormone tissues of your body, like the thyroid, to release their hormones. Right? So these blue and green triangles in the cartoon are these thyroid hormones, the T3 and T4, which are made inside of these cells, as peptides, right? So they are small proteins as well that involve iodine. So that's what these green eyes are over here, right? So I think you guys know iodine is an important part of good thyroid health. And that's because iodine is used to make things like these T3 and T4 thyroid hormones that then go out into different tissues of your body and control things like your metabolism or other aspects of your normal, well-balanced physiology. Right? So the problem is, if your adaptive immune system, for some reason, has B cells that make antibodies, so down at the bottom cartoon here is a yellow antibody that happens to be the right shape to actually bind to the TSH receptor on the surface of these cells, right? Now, let's pause here a moment, right? Most of the time, when we talk about antibodies binding to some sort of epitope target, we're talking about that in the context of, that antibodies has identified something that is a potential threat, it doesn't belong there, and then that antibody is going to now cause your immune system to somehow react against this target, potentially to eliminate it, causing damage, right, to cells in the body if you have antibodies binding to them improperly. In this case, though, I want you to consider what I started off this lecture talking about. Antibodies are free-floating proteins, right? Just like a cytokine message is a free-floating protein. Most cytokines would be a lot smaller than a typical antibody. Right? So if an antibody binds to the receptor that the small signaling molecule is supposed to bind to instead, it can actually improperly activate this receptor, right? Because by binding to it, the receptor kind of sees it as though something's bound to it, like the signal that it's expecting, but in this case, it's an antibody binding to it. And it'll go ahead and trigger the thyroid to continuously release these thyroid hormones, the T3 and the T4, even though the pituitary up in your brain is like, hey, what are you doing? Right? I didn't want you to do that, but too bad. You have all these antibodies that are stimulating the thyroid to do that. So as a result, you have uncontrolled release of hormones from the thyroid gland, and so you have changes in your metabolism, right? So a lot of times you have symptoms like an enlarged thyroid because it keeps getting overstimulated. So it keeps thinking that it needs to make more and more of these hormones. So as a result, you have more and more thyroid cells, right? You might end up with uncontrolled sweating, nervousness, and irregular heart rate, right? I think that's fascinating to think. That's an example of your immune system doing something that really the immune system normally doesn't do, which is to somehow trigger or stimulate receptors on other cells just because they happen to be the right shape. Another example of this is myasthenia gravis. So in myasthenia gravis, the IgG binds to neurotransmitter receptors at the junctions where nerve cells junction or meet up with, let's say, muscle cells, right? And so nerve cells that end up joining up with a muscle cell, like a skeletal muscle cell in your arms or in your legs, right? There's a little synapse there where neurotransmitter molecules have to be sent from the end of the nerve or the neuron and received on the other side of the narrow little synapse by the muscle cell having neurotransmitter receptor molecules on its surface. So if you improperly make IgG, that kind of gets in there and binds to these neurotransmitter receptors, right? Improperly stimulating them all the time, then you can imagine, right? You lose control over normal types of muscle movement. As a result, these receptors get inundated with Igs bound to them, and so you no longer get the proper messages going from the brain to the spine to your nerve to your muscle cells, right? So you end up losing the ability to control your muscles. So one of the typical symptoms of myasthenia gravis is loss of muscle responsiveness, right? Again, just because the adaptive immune system has improperly made an Ig that binds to an important receptor that's on the surface of other cells. In both of these cases, in a normal situation, right, these Ig shapes would never have been allowed in the first place, right? Because any naive B cell that came up with a cookie recipe to make these shaped Igs in the bone marrow probably would have either failed central tolerance training in the bone marrow and never be let out into the blood in the first place, or when it's let out in the blood and it's undergoing peripheral tolerance training, right? This naive B cell would have encountered these things, right? And have gone anergic. So normally a B cell like that would not be allowed to go to work and cause trouble like this. So again, a mistake is made. In this case, perhaps a failure of proper tolerance training. Okay. An important concept with autoimmune disease is that even though autoimmune disease can start off with one seemingly small mistake, right, like an Ig that improperly binds to some epitope that is part of some common common molecule or common surface feature of the cells in your body, right? The real problem over time happens when recognition of multiple different epitopes begin to kind of spread. In other words, even though the initial mistake is maybe one specific epitope being recognized, right, over time, right, that list of epitopes that are being recognized can broaden and expand. How does that happen? It happens because, remember, when an immunoglobulin on the surface of a B cell binds to some target, remember, IGs can bind anything, right? It could be a protein, it could be a carbohydrate, it could be a lipid. It just has to be the right shape to fit. But remember, when we talked about the antigen coming from something like a pathogen, right, in our sort of real conventional sense of our immune system should be fighting, infections, right? We said that oftentimes the thing that the Ig ends up binding, even if it's a carbohydrate or a small piece of protein, it's usually a part of some chunk of bigger piece of a pathogen, right, that has other types of proteins and carbohydrates and stuff associated with it. And that B cell will end up taking all of that in because it's binding to its Ig, and then it'll be presenting different types of peptides, potentially to other T cells that are coming to check it out. So this can cause then, right, a spreading of different things being recognized. Look at these cartoons here. On the left, you have a situation where you have multiple, so in this case, 4 B cells. The yellow cells in all the corners are 4 different B cells, all of which, for some reason, failed proper tolerance training. And their IGs on their surface, whether it's the yellow Ig here, purple one, green one down here, or orange one over here, are the right shape to be able to bind a normal body signal. In this particular case, the signals that they're binding to are things that are coming from inside of cells. Right. So let's look. For example, this B cell on the upper left-hand corner, right, its immunoglobulin is the right shape to bind the histone 1. Histone 1, of course, is one of the histone proteins that combine with other histones, like histone 2, histone 4, to help assemble nucleosomes as part of chromatin inside the nucleus of your cell, right? And around which the DNA, your DNA, is wrapped up to make chromatin. Right? So, of course, histone 1 should be something that B cells, right, should be trained not to freak out and be sensitive to. But in a situation where you don't have good self-tolerance training, maybe you have this B cell and its yellow antibody picks up on histone H1. Now you might say, wait a minute, this is a nucleosome with DNA wrapped around it. What's it doing wandering around outside of a cell? Hey, think about it every day, right? You have billions of cells in your body that are kind of turning over and being recycled, right? And part of recycling is if they undergo apoptosis or whatever, or they just get damaged, there's going to be the shedding of certain types of cellular components that normally are contained inside of the cell, maybe even inside the nucleus, right? So that normally immune cells on the outside of this particular cell wouldn't necessarily even have access to it. So in this case, you have some shed nucleosome structures with some DNA, and this B cell picks it up, right? So it's going to take the whole thing in, and it's going to present all the little peptides that come from the red H1, the blue H2, the pink H4, histones, whatever. In this case, right, so it's loading the red dot onto here on MHC2. And here you have, again, due to some sort of failed self-tolerance training, a CD4 helper T cell, right, whose T cell receptor is the right shape to bind to this peptide that comes from histone H1. Right? So this then represents a pairing of B cell and T cell that could be the start of trouble. Right? Notice over here, this B cell, it has a purple Ig, right? And it also improperly picks up on these nucleosome structures, but in this case, it's actually binding to DNA, right? That sounds really kind of screwed up, right? You're in big trouble if your immune system actually makes immunoglobulins and antibodies that bind to DNA. But that's what's happening in the cartoon here. The important thing here is what? Once again, this whole nucleosome complex gets brought into the B cell and it gets processed. So you're going to have some of the red dot, that histone H1, right, end up being presented by this B cell as well. And so once again, it activates that helper T cell, right? And so over time, you can imagine that if this keeps happening, right, you'll get more and more T cells that are activating against other components that may be part of the nucleosome here. The cartoon is just showing on the bottom here that here you have a B cell that binds to the ribosome, right? The ribosome that's translating RNA, right? So in that case, it's going to present different peptides that come from the ribosome, none of which would be the H1 histone. So therefore, this particular helper T cell wouldn't care. Right? On the right is another scenario, right, of this epitope spreading, where you have the B cell in the middle now, right, and it's pulling in something like a nucleosome because its Ig binds to H1. But of course, it presents not just the red dot that comes from the H1 histone, but the other colored dots as well, representing all the other stuff that it's pulling in, right? And so it's presenting all this stuff on MHC2. Because remember, MHC2 on the surface of a B cell or any professional antigen presenting cell, right? They're just presenting all kinds of peptides. But in this case, you have 4 different T cells that are coming and potentially interacting with this B cell. So you might end up activating that H1 specific T cell, H2 specific T cell, H3 specific T cell, but you're not going to get into trouble activating a ribosomal protein specific T cell. Right? Over time, the list of epitopes then would spread as more and more different adaptive immune cells that each recognize different specific targets, right, all get kind of brought in as these targets become more spread out. Okay, so let's now take a look at a couple of other examples of type 4 T cell mediated disease. In particular, type 1 diabetes and rheumatoid arthritis. So let's start with rheumatoid arthritis. So you guys know what arthritis is, right? Because we all have relatives, an auntie or an uncle or our grandparents, right, who say they have arthritis, right, and their joints hurt. So there are different types of arthritis, because arthritis, the name itself, really just means that there is some sort of inflammation of joints, right? So this inflammation could be caused by many different reasons. Just normal wear and tear and being old, right, can lead to arthritis, right? As the cartilaginous regions of our joints just kind of get worn down and are not sort of replenished or rejuvenated as well as they used to when we were young. Rheumatoid arthritis is a particular type of arthritis, though. This is not your everyday, just because you're old, your joints hurt, right? This is actually an autoimmune condition. So the name rheumatoid arthritis comes from rheumatic, right, which is very ancient Greek for rheuma, meaning body fluids, right? Because again, back in ancient times, right, I think medicine was very sort of focused on this idea of different types of fluids and spirits and things like that in the body. So rheumatic arthritis then implies that it has something to do with something in the system that's actually causing this arthritis, right? And so it affects about 1 to 3% of the US population. So just by that, you know, we're not talking about just every day, like I'm old and my joints hurt, right? This is something very specific. And again, in a common theme, as with other autoimmune diseases, rheumatoid arthritis impacts women about 3 times more than men. And it starts at an early age, right? So we're not talking about senior citizen arthritis. We're talking about something that begins between age 20 and 40. Rheumatoid arthritis leads to very painful swelling of the joints. And I think many of you are familiar with this, where the joints become so severely inflamed that over time, you actually get malformation of the joints. Because this is actually damaging the bone inside of the joints of digits like fingers and toes, right? And if untreated, you can actually get so bad that the individual loses ability to use that particular appendage. So this is a type 4 autoimmunity, meaning this doesn't have to do with antibodies. Type 4, right, has to do with T cells making a mistake. Let's look at these cartoons. So in a normal joint, pictured on the left here, you have your 2 bones, you know, so these might be the tarsals in your feet or the phalanges in your fingers. I think I got that right. I might have just revealed that my anatomy is really bad. But anyhow, right, so the finger bones, where they join up, of course, your knuckles, right, you're going to have bones meeting bone, but the ends of the bone are covered with this sort of slippery cartilage to allow them to have some shock absorption. And when they rub against each other, they're not actually grating against each other. So the knuckles are contained in sort of this sac. So you have a tough fibrous sort of capsule surrounding the joint, and it's filled with synovial fluid, right? And then around it, you would have things like your muscles, tendons, etc. In rheumatoid arthritis patients, if you look at their knuckles in their hand, for example, in their fingers, what you see is a lot of inflammation in the area. So the capsule is basically really inflamed, and you see the erosion of bone tissue. Okay. So what's going on here? Rheumatoid arthritis is a mistake of T cells. Right? The actual start of rheumatoid arthritis does not have to do with antibodies, but when things get progressively worse and worse and worse, eventually you can end up having things like immunoglobulins or IgGs produced against the various sort of normal antigens that are found inside the joint. So how does this happen? So it's a really fascinating story because there's definitely a genetic component to this. Right? And you might say, well, I don't really understand. How could there be a genetic component to developing things like, overinflamed knuckles, right? That just sounds kind of weird. But it has something to do with the immune system making a mistake, right? So what would be the genetic component of that? There's a particular enzyme that's made by the tissues of our body that's called peptidyl arginine deaminase, or PAD. What this enzyme does is that it removes arginine amino acids from short peptides, right, as part of processing peptides. Once the arginines are removed, we say these peptides are citrulinated. Right? This is a fancy way of saying that these are peptides that have been processed in this way to have different amino acids removed. So PAD, this enzyme that does this, is usually expressed in tissues that are damaged. Because in damaged tissue that needs to be recycled, part of that healing process is removing the damaged tissue, right? And so that's not surprising then you'd have enzymes like this that are participating in the breakdown of proteins and things and peptides as part of the recycling. Now, normally that happens if you have an injury somewhere, right? So it's just kind of a passing transient thing that the tissues make this PAD enzyme. And then once you're healed, you don't need to make it anymore. But think about conditions where you constantly damage the tissue over and over and over again, right? Chronically, like, for example, smoking. So heavy smoking increases the damage in your lungs and the production of this PAD enzyme in your lungs, right? Resulting in your immune system in heavy smokers or chain smokers, right, constantly having higher levels of this PAD enzyme production in lung tissue, means their immune systems see more examples of these processed citrulinated peptides from damaged tissue than a typical non-smoker would see. As a result, what you're doing is you are setting the stage that if that person then has naive T cells that recognize these citrulinated peptide shapes, there's a greater chance in a chronic heavy smoker of activating those naive T cells compared to in a non-smoker's body, right, where you only occasionally have PAD and citrulinated peptide present when you get injured. Now, that means over time, many years of heavy smoking, the person builds up, right, a population of these T cells that are now patrolling around looking for these citrulinated peptides. Right? And then what happens later? As you begin to have your joints naturally age, right, and begin to kind of, have some damage, then those are the locations where you now have many of these citrulinated peptides, right? Because the PAD enzyme goes to work in these tissues and makes those citrulinated peptides. And if you have these T cells that are on the lookout for that, what do? They start attacking your joint tissue, right? All the joint tissue cells that are expressing or displaying these processed peptides. So this gets back then to what we discussed last time, that oftentimes the genetic component of autoimmunity has to do with does your genetic makeup in terms of your MHC or HLA polymorphisms that make your MHC platforms on your cells, right? Do you have a particular genetic background of polymorphisms that make particular shaped MHC platforms that might be better at displaying something like a peptide that gets you in trouble compared to somebody else's body who, because of different polymorphisms, different family lineage, right, they don't have those same shaped MHCs. So their cells don't present that peptide as much. And therefore, the risk of their immune system making a mistake against that peptide is much lower. In this case, MHC class 2 polymorphisms involving the HLA-D protein R4 allotype favor the binding and presentation of these citrullinated peptides. So again, quick reminder, right? What we're saying here is that all of our cells in our body on the surface make multiple types of MHC proteins, right? So that we can present many different examples of things that are coming from inside. And I said before, let us pretend, right, that in this case, the genetic makeup of this person means they have all three of these MHC platforms on their surface, the Chinese guy, the Roman gladiator guy, and the stuffy, stuck-up British guy. I shouldn't say that. But anyhow, so the point is, right, because of the genetic makeup, you can present on this cell a good job of presenting the chicken head that comes from inside, right? But if you didn't have that genetic makeup, maybe the cells in somebody else's body only presents the wing and the foot. Right? So let's pretend that the chicken head is these citrullinated peptides. That means if you have a genetic background that your cells have this particular, Chinese guy over here, MHC, right? Your cells do a better job of presenting the citrullinated peptides. That means there's far more likelihood of your immune system, your T cells, making a mistake and seeing it compared to somebody else, right? So this has to do with the HLA DR4 allele. So in other words, this particular MHC here, right? And I believe it's the beta chain part of it. So let's look at this. All right, so here is the MHC class 2 again. There's an alpha part and a beta part. Right? And so these are coded for by different genes. And so what we are saying is that depending upon your genetic background, the shape of your HLA DRB or DR beta chain over here might be slightly different. Right? If we were to look at this region, remember this is the cleft that actually holds the peptide. If we were to look straight down at it, we would see this. Right? So we see the two sides of the cleft, and in yellow and red is what we see on the left here, now looking down. And what we're doing is we are pretending that these red dots represent the positions of particular amino acids in this chain, because remember, each of these chains is just a polypeptide chain, where we see variations in the human population. In other words, each one of these locations by the red dots is where a different polymorphism has been identified and cataloged. Okay. On the right, you can see here, this table shows you that in the HLA-DR beta chain, at amino acid positions 67, 70, and 71, these seem to be the critical ones involved in the risk of developing rheumatoid arthritis. So let's pretend, for argument's sake, I don't know over here which of these red dots really are 67, 70, and 71, right? But let's just pretend. Let's just say maybe this is 67 over here, this is number 70, and this is number 71. Those three amino acid positions, right, that different people have different amino acids based upon their genetics. So you can see here are 5 examples of polymorphisms having to do with this DRB, HLA DRB1 position, RB1 polymorphism, right? So what they're showing you is that if you have a lysine, a K at 71, right, they're putting this in blue because that means your risk of developing rheumatoid arthritis, right, is a bit higher than the normal population. Likewise, if you have an arginine, an R at position 71, right? Why? because let's go back to this cartoon. Let's pretend this is amino acid 71 over here. What it's saying is if you have a lysine or an arginine in this position, right, the shape is going to be slightly different than somebody else who has a different amino acid at that position. And if you go down here, that means maybe your MHC2 hangs on to citrullinated peptides a lot better and is able to present that as the chicken head. And therefore, you're at a higher risk of T cells coming along and seeing that and making a mistake. Right? It doesn't mean you're guaranteed to be autoimmune, right? It doesn't mean you're guaranteed that somewhere along the line a T cell will make a mistake. It just means your risk is higher of that happening. But this is the interesting thing. It works the other way too. In people who have amino acids like a glutamate here in 71 and an aspartate, in position 70, like a D and an E, right? These are actually protective. Why? Because the shape of their MHC is really bad at hanging onto the citrullinated peptide. That means on their cells do a lousy job of displaying this problematic peptide to the immune system. So they would be like, if we go back to the chicken cartoon, right? They don't have the guy on the left presenting the head. So their risk of having T cells accidentally making that mistake against that peptide is much lower, right? So I think that's interesting, right? Your polymorphisms, depending on what you got, can either put you at higher risk or even be protective, all based upon what your immune system ends up seeing from your cells that are presenting things. Importantly, infections may actually trigger or be the start of autoimmune disease. So this is kind of scary. Right? Consider that if a pathogen infects you and your body properly selects for things like Igs or TCRs that recognize, different potential antigenic features of that pathogen, as we've learned, that's the way it's supposed to work. Right? So you'll select for a bunch of B cells or a bunch of T cells that all recognize different features on this pathogen. And hopefully, right, once you've survived that infection, you'll have memory cells that stick around. So if that pathogen comes back again, hey, you already have those selected cells that can go to work right away. But what if those antigen shapes on the pathogen, right, just by chance look a lot like certain normal shapes inside of the body? I don't think it's hard to imagine that this could happen, because after all, when we're talking about little antigens, we're just talking about molecules with different shapes, and the actual little epitope, the specific shapes that something like an antibody would bind to, right? It's a pretty small three-dimensional feature. So in fact, I think it's more likely than not that you would have examples of other shapes in your body that kind of look like the pathogen antigen. After all, we learned that that's why we already produce things like immunoglobulins against other people's ABO blood types, right? Because, these are shapes that are very similar to things that our immune system has evolved to recognize in bacteria, right? These lipopolysaccharides in bacteria, are very similar to the glycolipids on the surfaces of red blood cells. Right? So just with ABO blood type rejection, the cells in your body then might end up being marked for destruction by these Igs that you ended up selecting for as part of fighting against the bacteria. So let's consider this. On the left part of the cartoon, you have a bacterial infection. Maybe this is streptococcal bacteria, like the kind that give you strep throat. On its surface, it has red triangle antigens, green circle antigens, right? And so as part of beating the infection, you've selected for B cells that got really good at making Ig shapes, that when the B cells are told to turn into plasma cells, they make a lot of antibody that bind to either the red triangle or the green, circle. So maybe the green circle shape is something that's very rare inside of our bodies normally, right? But what if the red triangle shape is actually something that you do find in other normal cells of your body? For example, what if the cardiac tissue that lines the chambers of your heart express a particular protein that's an orange triangle, right? So it's slightly different than the red triangle, but the point is, right, they still make it a triangle in the cartoon because it's really close in shape. It's likely then that these yellow antibodies that you've produced as part of fighting the Streptococcus bacteria, and now you have memory cells that make them, as they're flowing through your blood and through your heart, they might start binding to the orange triangle shape. Right? So this would be just an awful kind of coincidence if the red triangle on the bacteria and the orange triangle on your normal body cells are close enough that this case of mistaken identity can happen. Right? Because normally, right, if it were not for the infection in the first place, your bodies, your developing B cells, would have been self-tolerance trained, right, to not allow any B cells that bind to the orange triangle in your heart tissues, right? But because you had to select for B cells to fight against the bacteria, and you selected for ones that bind against the antigen on the bacteria, right? Now, suddenly, right, you have this situation where you have these antibodies binding to that self-antigen. Right. So here are some examples of different autoimmune conditions that we believe now have strong links with starting from some kind of pathogenic infection. So things like a rheumatic fever, this is the example that I just showed you with the heart. We now know that there seems to be some link between having streptococcal infection and then the risk of developing rheumatic fever afterwards. Other examples you can see here, even certain types of food poisoning can lead to things like arthritis-like situations where autoimmune cells begin to attack your joints. This one down here is really interesting, and we'll sort of end our discussion today with this example. Coxsackie virus. Coxsackie virus, there's now a strong link, we know, between getting Coxsackie virus infections and the development of type 1 diabetes. So type 1 diabetes, what is it? There are two types of diabetes type 1 and type 2. Type 1 is often referred to as insulin-dependent diabetes. Right? This is the one where, I think people typically think of diabetes as you have to give yourself injections of insulin. Right? Why? Because insulin is an important hormone that's made by the cells of your pancreas, in particular, these beta cells in the islets of your pancreas. When you eat a big meal and you start digesting food, you're going to have a lot of sugar from the food that you're eating going through your blood. And your body's tissues need to get a signal that tells all of the cells in your body, hey, we got lots of sugar in our blood, go ahead. Start taking a lot of this glucose out of the blood and bring it into the cell so you can start using the glucose for energy. Right? So that signal is the insulin. When we have a big meal, our pancreatic islet cells begin to make lots of insulin, and the insulin flows around our body, and all of our tissues, right, receive the insulin, and that's the signal to start taking sugar out of the blood and using it. Right? So type 2 diabetes is different. Type 2 diabetes happens when, due to things like diet and lifestyle, you actually end up having tissues in your body that no longer respond to insulin flowing around in it. Okay, so it's two very different types of diabetes. Type 1, or the insulin-dependent one, has to do with the immune system, for some reason, improperly recognizing these pancreas cells and attacking and killing the pancreas cells. And therefore, the body no longer makes insulin in the first place. So we call this juvenile onset diabetes because type 1 diabetes is primarily in kids. Right? Type 2 diabetes is usually in adults, right? Like once you're past middle age and, if you're like me, right, don't get enough exercise, start putting on weight. My physician's always warning me, hey, you're pre-diabetic. You're right on that border. You got to exercise, you better eat right, Okay. So type 1 diabetes, right, is caused by CD8 cytotoxic T cells attacking the child's own pancreas beta cells, right, and destroying those beta cells so that the child can no longer make insulin. And without insulin, the child's tissues throughout her body never get that signal to take the sugar out of the blood and use it. And as a result, the sugar levels can build up in the blood to dangerous levels, right? That can actually send the child into shock, or worse, actually cause death. Okay. So now we know that there is a link between Coxsackie virus and type 1 diabetes. So Coxsackie virus, what is it? You're like, that's a really weird sounding name, right? It's actually a very common virus. Coxsackie virus is the cause of what we call a hand, foot, mouth disease, right? I think some of you, the moment I said that, you're like, oh, I know what that is, right? Hand, foot, mouth disease is something that little kids often get from preschool, right? They go to preschool, they're playing with all their friends, and they pass around this Coxsackie virus, and they end up with hand, foot, mouth disease, and they have like little blisters on their mouth and on their feet, you know, on their hands, right? So for some reason, right, as the child's body, her adaptive immune system learns to fight the Coxsackie virus infection, there is some antigen presented by the Coxsackie virus that must look a lot like some kind of antigen that is normally made and presented by the pancreatic islet cells. So that after the child beats her infection, and I should say that the Coxsackie virus itself, it's not a lethal infection, right? The kids just have these blisters and things, you know, but every so often, right, a child's immune system might make that mistake. They've selected for immunoglobulins and T cell receptors that recognize these antigens from the Coxsackie virus. And now, even though the infection's over, they're starting to now mistakenly see something similar from the pancreatic cells, and the child's immune system goes to work and starts destroying her own pancreas cells, right, which causes type 1 diabetes. Okay. So let's look at this. Just like with the rheumatoid arthritis, the genetic component is involved here, right? Because whatever this mysterious peptide is that's being presented by the pancreatic islet cells, right, it's being presented on MHC2. So that means children with genetic backgrounds that present more of this peptide, right, are at higher risk of developing type 1 diabetes than children with a different genetic background whose pancreatic cells do a lousy job of presenting this peptide, and therefore they can sort of avoid getting into trouble, right? So again, it has to do with the HLA D locus, the beta chain, and just like with the rheumatoid arthritis, there are 4 amino acid positions that seem to be very important, right? And depending on what block of HLA gene polymorphisms you inherit, this can be protective. So for example, if you have these amino acids in these 4 positions, right, I guess it means your pancreatic cells do a bad job of presenting whatever this mystery peptide is, right? So you have a very low risk of T cells making a mistake and seeing it there. And that goes all the way to high risk. So children who are born with this genetic makeup and have these amino acids in this position, their pancreatic islet cells, right, do a very good job of presenting whatever this mystery peptide is. And so they are at a much higher risk of developing that mistake, that mistaken identity. Doesn't mean they're guaranteed to develop type 1 diabetes, right? But it sets the stage for that mistake to be made, right? And that mistake, now we know, can be triggered by something like the hand, foot, mouth disease. So quick story to wrap up. My cousin John, he has a daughter, and several years ago, I think this is like 6 years ago, when she was like 4 years old, 5 years old, one day they brought her home from preschool, and my cousin's wife, Kathy, noticed that their daughter was just laying on the floor, really tired, right? And her daughter was complaining about being super thirsty. Fortunately, Kathy is a family practitioner, family physician. So right away, right, she knew that these were symptoms of potentially type 1 diabetes. And so she rushed their daughter to the hospital, right? And fortunately they did, because her sugar level in her daughter was so high that they said that had they not realized to bring her to the ER, their daughter might have slipped into a coma. Okay. And it turns out their daughter developed type 1 diabetes. And she had to learn at age 4 how to give herself injections of insulin. Right? Really sad. Right? Now she wears a little pump, right? That can control the injection of insulin into her body, so she doesn't actually have to give herself shots anymore. But she has to always constantly monitor her blood sugar levels, and because she's always wearing this little pump, she cannot engage in normal types of activities like sports or swim or anything like that. It's really sad. I think what's really sad and kind of poignant about this is, sometime later I was talking with Kathy, the mom, about it, and she actually told me that, yeah, right, 3 months before that day when their daughter nearly went into a coma, right, her daughter did catch hand-foot-mouth disease from preschool, right, and she was sort of homesick for a few days, but she got over it, and they just thought that was the end of it, right, like normal kids would just get over it. But in her case, that mistake was made by her adaptive immune system, right? And there is no cure for type 1 diabetes. So as things stand right now, right, for the rest of her life, until we find a cure, her pancreas is always trying to make new beta cells, right? But as they're being made, they're being destroyed by her own immune system. So she can never make the insulin that she needs, and she has to get it from an outside source. So it's quite possible that the genetic combination of my cousin and his wife may have been one of these high-risk genotypes, right? And her daughter has to live with that. Okay, so let's go ahead and stop there today.